Hey guys, today we're going to be going over another lesson about Punnett squares. This is actually going to be the last type of Punnett squares that we're going to work on. So far we've done monohybrid, which is the stuff that you did in grade 9, then our dihybrid squares, incomplete dominance, codominance, multiple alleles, and then today we're going to finish it off with sex-linked traits. Okay, so sex-linked traits. For this, we're going to be talking about what sex-linked traits are, why they tend to affect more males than females, and then all of the different phenotypes and genotypes that you can get with sex-linked traits. Then, of course, we're going to do some Punnett squares, identify our genotype and phenotype ratios, and then lastly, we're going to talk about the work of Thomas Morgan and Barr bodies. So sex-linked traits are traits that are determined by genes that are located on the sex chromosomes, so either the X or Y chromosomes in humans. Unlike most traits, which are on the autosomes or the non-sex chromosomes, these traits behave a little differently because, for males especially, they don't have homologous sex chromosomes, and it is the only pair of chromosomes in the human body anyway, that are non-homologous. The X chromosome is much larger than the Y chromosome. It contains way more genes. And so because of that, most sex-linked traits are on the X chromosome. There's just more traits on the X chromosome than on the Y chromosome. If you do have a sex-linked trait that's on the Y chromosome, we call it a Y-linked trait. All others are X-linked traits. Most sex-linked traits are X-linked recessive, so a recessive trait on the X chromosome. And because of that, most sex-linked traits are more common in males than females. The reason for that is that females have 2 X chromosomes, so in order to get the recessive X-linked trait, they have to have 2 copies of it. Whereas with males, because they only have 1 X chromosome, as soon as they get 1 copy of an X-linked recessive trait, they have that trait. Yeah, only women can be heterozygous for an X-linked trait, in which case they still won't have it as long as it's a recessive trait. That'll maybe become more clear when we actually look at the different genotypes and phenotypes. X-linked dominant traits are much less common, but they affect females more because if the X-linked trait is dominant, you only need 1 copy of it to get that trait, and women have twice as many chances of getting an X-linked dominant trait because they have twice as many X chromosomes. Okay, so with sex-linked traits, there are 5 different genotypes, and each one has its own phenotype. So the chart below kind of summarizes this. So the first 3 genotypes are for females only because they are all XX. With sex-linked traits, you always have to write the sex chromosomes, and then you write the trait as a superscript. So you can have the first genotype here, which is a female that has both of the alleles as the dominant one, in which case she is perfectly normal and does not carry the X-linked recessive trait. Or you can have a heterozygous female, in which case she is what's called a carrier. So she carries the recessive trait, but she doesn't actually show it. So she doesn't actually have any of the physical symptoms, but she does carry that trait so she could pass it on to her children. And then if you have a homozygous recessive female, where she has 2 copies of the X-linked recessive trait, then you will have a daughter or a female with that trait. And then there's only 2 possible genotypes for males. Those are the last 2. Either it's a male that has the dominant version, in which case he'll be perfectly normal, or you have a male that has the recessive version, in which case he'll have that recessive X-linked trait. Okay, so only females can be carriers because you have to have one dominant and one recessive version to be a carrier, and in order to do that, you need 2 X chromosomes. Okay, so a carrier is someone who has the allele for a trait but does not express it. Carriers aren't specific to sex-linked traits, though we do use the term for sex-linked traits more, but really anyone who is heterozygous for a recessive trait is a carrier. Anytime they have the recessive allele but the dominant trait, because they have a dominant version and a recessive version of that gene. Carriers can only exist for recessive traits. Because a dominant trait will never have an allele that's present but not expressed. As soon as a dominant allele is present, that trait is expressed. In X-linked traits, only females can be carriers because males only have one copy of any X-linked traits. And carriers are also sometimes just referred to as heterozygous females. So we're going to be going over a sex-linked Punnett square. Because we're only going to be looking at one trait at a time, this is just going to be a little 2x2 square. And then we're going to look at the genotype and phenotype ratios. So let's work through the first example together, and then you guys should try the second one on your own before coming back for the answer. Okay, so for the first example here, it tells us that colorblindness is an X-linked recessive trait. It says that the father is colorblind and the mother is a carrier. So that means that the dad, who's going to be XY, if he is colorblind, then he has that colorblind trait on his X chromosome. And then for mom, she's going to be XX, so she is a carrier. Okay, in this case, I am using the C that looks like a cent sign to be the dominant version, which is just normal sight, and the little c to represent color blindness, which is the recessive trait. Okay, and then we are going to make our Punnett square. Again, this is just a little 2x2. We're just going to separate the sex chromosomes with their alleles on them. It doesn't matter which side the mom goes on and which side the dad goes on. And then we will fill out our box just like normal. Again, when you have 2 alleles like this, we always write the dominant one first. Okay, so that's what this should look like. And then to look at our genotype ratio, so we could have, again, there's 3 different possible genotypes for females, only 2 for males. We can either have homozygous dominant, heterozygous, or a carrier, or a homozygous recessive female. And then for the male, either he can have the dominant or the recessive trait. So in this case, we have a 0 homozygous dominant, 1 carrier female, 1 homozygous recessive female, 1 dominant trait male, and 1 recessive trait male. Okay, so the ratio is going to be the same for the genotypes, which is at the tops there, as it will be for the phenotype. So the phenotype, we have 1 carrier female, we have 1 colorblind female, we have a normal boy, and we have a color blind boy. Those are our 4 different phenotypes that have shown up, we have no totally normal girls. Okay, so try the second one on your own and then come back for the answer. Okay, this one's a little bit trickier. I'm going to use a capital H to represent the dominant healthy version and a lowercase h to represent the recessive hemophilia version. It starts by telling us that we have a non-hemophiliac woman. So non-hemophiliac woman, either she could be dominant, homozygous dominant for the healthy trait, so 2 capital H's, or she could be a carrier. Either way, she won't have hemophilia. So we know that for sure she has 1 capital H, since she doesn't have hemophilia, but we don't know what her other X is carrying. It then tells us that she marries a healthy man. So in order for the man to be healthy, his X has to have that capital H on it. It also tells us that the woman's father had hemophilia. So the woman's father, I've written up top here, he must have had a little h. Now this woman would have got one of her X chromosomes from her mom and the other X chromosome from her dad. That's how genetics work. And so because the dad only has 1 X chromosome, he has to give that one to his daughter, which means that his daughter, this woman that we're talking about, is a carrier for hemophilia. So now knowing that, we can do our Punnett square. And it'll look like that when we have it all filled out. So then our ratio for genotypes is going to be 1 homozygous dominant female, 1 carrier female, no homozygous recessive females, and then for our 2 males, our 2 male genotype options, we'll have 1 of each. Which means as far as phenotype goes, we've got a normal healthy girl, we have a carrier girl, we have no hemophiliac girls, who I'm calling a hemo girl for short, and then we have 1 healthy boy and 1 hemophiliac boy. Okay, so that's it. That's the answer you should have. Okay, so that's how you do a sex-linked Punnett square. We're now going to talk about Thomas Morgan, who was kind of the pioneer when it came to understanding sex-linked traits. So Thomas Morgan was an American biologist who experimented on fruit flies to learn about sex-linked traits. He chose fruit flies because they have a really short gestation and maturation period, which means that you can like have a bunch of fruit flies born and they'll like turn into adult fruit flies really quickly. They also produce a large number of offspring at once, and you obviously don't need a lot of space to store and experiment on the fruit flies. You can also tell the males and females apart really easily, which is important when you're doing any sex-based genetic research. So this was back in 1910 that Morgan found amongst his fruit flies that one of the males had strange white eyes. Usually they have red eyes. And so he crossed that white-eyed male with a red-eyed female, and all of the offspring, which is the F1 generation, had red eyes, which meant that the white eyes were recessive. Then he picked a male and female from that F1 generation, who, remember, all had red eyes. And in their offspring, the F2 generation, all of the females had red eyes, but only half of the males did. The remaining half of the males had white eyes. So because this recessive trait was only showing up in males in the F2 generation and not in any of the females, Morgan was able to tell that the gene for eye color in fruit flies is on the X chromosome and therefore an X-linked recessive trait. So based on that, what was the genotype of the P1 parents that he first crossed in that experiment? Then I want you to complete a Punnett square in order to determine the genotype ratio of the F1 generation, and then find the genotype ratio of the F2 generation. So go through and answer these questions and then come back and I'll let you know what the answers are. Okay, so in terms of what the genotype of the parents were, we know for sure that the white-eyed male was carrying the recessive trait. So if we say that capital R is for red eyes because it's dominant and lowercase r is for white eyes, then the male parent would be XY because he's a male and would have a little r on his X chromosome. And then the female could have been either homozygous dominant for red eyes or heterozygous. But because all of the children ended up with red eyes, we know for sure that she was homozygous dominant. If she was homozygous recessive, she would have passed on that recessive trait to some of her sons at least, and they would have had white eyes. So this is kind of what that should look like. So again, the male is going to have the lowercase r, the mum has two capital R's, and the reason that we know that she wasn't a carrier is because if she had a lowercase r, some of her sons would have inherited that X with a lowercase r from her. The sons all get the Y from the dad and the X from the mum. So if she had a lowercase r on her X, some of her sons would have got it for sure. And then same with the daughters. The daughters will all get the X from their dad, which means that they would have at least one lowercase r, and yet none of the daughters had white eyes, so they all got an uppercase R from the mum, which again wouldn't have happened if the mum was a carrier. So then we're going to complete a Punnett square between those two parents and see what the genotype and phenotype ratio is for their children. Okay, so here were our parents. When we do our Punnett square, we should get something that looks like this. So as you can see, it is a 2-to-2 ratio between our carrier females and our healthy males. These two are our two healthy males. These two are our carrier females. So a 2-to-2 ratio or a 1-to-1 ratio between those two options. Next step is to take one of these females and one of these males and cross them between each other. Right now, these four are our F1 generation. When we cross a male and a female, that's how we get our F2 generation. So let's do that. So I'm going to take my healthy male and my carrier female and cross them. And it'll look like this. Now what we get from that is a 1-to-1-to-1-to-1 ratio between our totally normal female, our carrier female, who's this one down here, our normal healthy male, And then down here, which would be our one white-eyed male. So both the normal female and carrier female both still have the red eyes because they have that capital R. Our normal male still has the red eyes. And then we have this one guy with the white eyes. So that matches up with what Thomas Morgan said. He said in the F2 generation, all of the females had red eyes and half of the males had red eyes, while the remaining half of the males had white eyes, which is exactly what we're seeing here. Okay. So that matches up pretty closely with what Morgan saw. Like I said, if this was not a sex-linked trait, then the males would have an equal chance of the females of having the red eyes or the white eyes. So if this was an autosomal trait, we wouldn't see that only the males ended up with white eyes and none of the females. It is only because this is a sex-linked trait that we have the males much more affected than the females. Okay, let's go on to our last topic for this slideshow, something called Barr bodies. So Barr bodies is a dark spot of chromatin located in the nucleus of female mammalian cells, so female mammals. Every female somatic cell has 2 X chromosomes, and because 2 X chromosomes is unnecessary, it's redundant, one of the X chromosomes is always inactivated. The inactivated X chromosome then condenses into a tight little ball called a Barr body. That means that only one allele of each gene on the X chromosome is expressed in each cell. It is random which of the 2 X chromosomes is inactivated in each cell. About half of the cells will inactivate one of the X's, the other half of the cells will inactivate the other X's. In the case of X-linked recessive traits, that trait will be expressed in about half of the cells then, because as soon as an X chromosome becomes inactive, that means that its alleles no longer have an effect. So for instance, if you have a carrier female and the dominant trait is inactivated because its X is inactivated, then the recessive trait will actually show through for that cell. Now, the person still won't express the trait as a whole because about half of their cells would still be showing the dominant trait. But there are some species where you can actually see which cells inactivate which X chromosomes. So for instance, in a tortoiseshell coat-colored cat, this coat color happens in heterozygous females. And essentially what's happening is that half of the X chromosomes code for like a black color, and the other half code for this kind of like tannish color. And so originally this cat would have 2 X chromosomes, so these are the 2 X chromosomes, and one would have the allele for the tan color and one would have the allele for the black color. And then in some of her cells, the black colored X chromosome becomes the Barr body, it becomes inactive, and in that case, the tan color shows through. And in other spots, The tan color will be on the inactive X chromosome on the bar body, and then the black color will show through. And that's how you get this kind of tortoiseshell pattern. So again, for a lot of traits, you can't actually see where different X chromosomes are activated, but there are instances like this where you can, which I think is really neat. Okay, that is it for this lesson. I will give you more practice questions on sex-linked traits later on. Yeah, that's it for now.